Hello students, welcome to Capman Academy, and today we are going to talk about tDNA integration into the host cell DNA. So we already know about what is tDNA, right, and how it is being produced and how it is being translocated from the Agrobacterium cells into the host cell, right? So today talk is going to be about the tDNA integration into the host DNA, right? So we already know when the tDNA, it actually enters into the host cytoplasm, it contains super T complex, which consists of proteins like Vir-B protein, D4 protein, which are a part of type 4 secretion system. Other than that, they also contain D5 protein, Vir-E2 protein, Vir-E3 protein, and Vir-F protein, right? And together they all make super T complex, right? And they also have here D2 and tDNA nucleoprotein complex, right? So now let's talk about the integration into the host genome. So first of all, this whole super T complex by the protein Vir-E2 is being trafficked, or you can say transported by the ER endoplasmic reticulum towards to the nucleus, right? Why E2? Because previously we discussed about it actually protects the tDNA from the nucleases, right, which can degrade the tDNA, right? So as it actually reaches into the nucleus, this Vir-E protein, Vir-E2 protein here, it gets degraded. It here gets degraded, right? By what protein? By the action of Vir-F protein, which is already present in your super T complex, right? And this happens as the Vir-E2 protein, it interacts with the VIP1 protein, or this one is called VIP protein 1, right? So as it interacts with the VIP1 protein, this one leads to the activation of Vir-F protein, which actually leads to the degradation of both of the protein. It leads to the degradation of both proteins, which are your Vir-E2 and VIP1 protein, right? So now, in the nucleus, it's further, now it has your T complex without the E2, right? And it has D2 protein here, and it has type 4 secretion system, right? So they all proteins are actually gets protected. They all gets protected by what? By the E2 protein. And when the E2 protein is degraded by the Vir-F protein, so it actually released from the T4 DNA. So now we have here tDNA with the D2 protein and your type 4 secretion system, and it has Vir-F protein now. And this one actually complex is protected by Vir-D5 protein from the proteolysis, right? You need to remember when it actually enters into the cytoplasm, this is called super T complex. As it is being trafficked towards the nucleus by the endoplasmic reticulum, in that case E2 protein, it interacts with the VIP1 protein, which gets degraded by the Vir-F protein, right? And this Vir-F protein with the tDNA complex, it gets protected from the proteolysis by the Vir-D5 protein. So each and every Vir protein has some special function here. You need to remember that, right? Then the host DNA. This host DNA, it actually leads to the integration. It leads to the integration of tDNA into the genome by its own repair mechanism. By its own repair mechanism, right? In that case, this tDNA here, it gets integrated, in which 3' end of the tDNA, it contains the eukaryotic specific sequences. Eukaryotic specific sequences, right? Like promoter sequences. Promoter sequences. It also contains polyadenylation sequences or splicing sequences. So these all eukaryotic specific sequences in the tDNA are called your microhomologies. Microhomologies, right? Which means they are identical, or you can say homologous to the eukaryotic genome, which contains the homologic promoter sequences or other type of sequences which are needed for the tDNA expression in the host. So this whole machinery of the host DNA leads to the expression of the tDNA, right? After the tDNA expression, another step comes, which is the expression. What type of expression of tDNA will occur? So the tDNA expression, it could be of two types. It could be number one, transient, and it could be second, stable transformation. Transient mean here the gene transcribed without the integration. In this case, this integration part will not happen, and the gene is actually transcribed independently. Transcribed independently by using the host genome, right? Sorry, host machinery. And this will actually takes 2 to 4 days of the transformation, right? But this one, in this case situation, if the expression is transient, in this case, the tDNA will not be transported or transferred into the progenes. Whereas in case of stable transformation, in this case, DNA is actually gets integrated into the host, and it will also pass through the progeny. And this mechanism takes 10 to 14 days. So it depends on the expression, how the tDNA is being expressed. The genes are being actually transcribed. tDNA genes are being transcribed, right? Now, this expression of tDNA, it is important in the recombinant DNA technology, because tIDNA is here used as a potential cloning vector. Cloning vector, in which some part of the tIDNA is being disarmed, which are majorly the auxin and cytokinin expressing part, and they are, as we know, are responsible for the tumor induction. This part is actually removed from the tDNA, right? And in place of the auxin and cytokinin part, we actually insert our gene of interest between the tDNA borders, and we use it for the expression of our gene of interest, right? Not the formation for the tumor. So the total this Ti plasmid size, it could be from 100 to 800 kilobase pair, and according to that, it actually accommodate the DNA inserts, right? So with this, our Agrobacterium mediated gene transfer mechanism is actually being completed. In that we saw the first step is the chemotaxis, which occurs by the wounds into the host cells, which releases the phenolic sugars, and that signals were actually taken by the Vir-A and Vir-G, which act as a signal perception proteins. They leads to the second Vir-G expression, which finally leads to the tDNA production into the Agrobacterium tumefaciens cells. Right? So as the tDNA is produced here by the association of some Vir proteins, then it further moved for the transfer into the host cell, right? Where it enters into the cytoplasm, and by the help of some of the protein, it actually gets protected and then moved into the nucleus, right? Where it gets integrated. So this is how the Agrobacterium infects the plant host cells.